Hi, Dr. Gildow. How are you doing today?
I'm very good, thank you, and thank you for inviting me this
Thank
morning.
you for being here.
Yeah, glad to do it.
Yeah, so how did you first involve in plant virology?
How did I first become involved in plant virology? It's a long story.
Yeah,
and
talk.
involving my whole career back to the early days. I'll start with my background. I have a BS degree in zoology from Ohio University. Got that in 1970. And then I decided, I switched from being interested in animals to plants.
Oh,
and
that's
so I
interesting.
decided I had a botany class, and it was so interesting that I decided I wanted to know more about botany and plants. So I got a master's degree in botany, doing mainly plant tissue culture and cell biology type work, but I was also interested in just how plants grow and plant ecology and that sort of thing. And then I decided to go for a Ph.D., and I was accepted to Cornell University. And I went for an interview at Cornell, and I interviewed all the faculty to see who I might be interested in working with. At that time at Cornell, the student was accepted on departmental funds, and you could change advisors anytime you wanted to within 1 year.
So it's like a rotation?
Not even a rotation. You could work for one person, and halfway through, if you wanted to change, you could change. A mutual decision. And you were encouraged to do that. So I interviewed all the faculty and found out what they were interested in. And I'd almost decided to work with fusarium, the effect of bentolate fungicides on fusarium with Jim Aist. But during my interview visit, I had not visited one faculty member. He was traveling at the time, and his name was William F. Rocco. And he worked with aphids and viruses. So I was sort of required to visit all the faculty, so I went to see Dr. Rocco. And it was summertime, because I had moved to Ithaca to go to school. So I was there, enrolled, but I had no advisor, and it was summertime. And I visited him in his laboratory, and Dr. Rocco said, well, the only way to know if you want to do something is to try it.
Yeah.
So come and help me set up an experiment. So we went to his lab, and we set up a transmission experiment, a virus transmission experiment with different aphid species and so on. And he explained what he was doing, and I kind of helped him as labor. And then he showed me how to wash the cages and clean up, and left me to do that, to clean up, and he left. And 15 minutes later, he walked down the hall, put his hat on, had a briefcase in his hand, waved goodbye, and said, I'll see you in the morning.
Wow, so he knows.
And I thought, what did I say? I hadn't said I wanted to work with him at all, and I was a little confused. He assumed that I must have wanted to work with him right off the bat, or I had more time to look at things. So the next, I asked his technician, I said, what time should I be here in the morning? She said, well, he comes in at 7 o'clock, but if you're here at 7:30, that will be okay.
Wow, early.
And this is summertime.
Yeah.
So I was there at 7:30, and he took me in his office, and he outlined 3 different types of experiments that were very different, asking different questions. And he said, why don't we start these 3 experiments, and then we'll see which one makes progress, and then we can pursue whatever works of these 3 things. And I'm still sitting there thinking, what did I say to this man? I did not agree to work with him. I didn't ask him to be a student or anything, but I thought he was such a nice person. I felt bad saying no.
Yeah.
And also I thought I was always interested in insects because of my degree in zoology. I had a degree in botany working with plants, and this involved the viruses, which I knew nothing about. But I thought this was, with these 3 systems, there had to be something I would like, and it would be very interesting. And so I decided, I'll give it a try. So I'll try it. So I became a student, and the research I did became successful after a while, and we discovered something very new and unusual. It became a big question, and so it really built my interest in virology and working with insects and vectors and that sort of work. And so that's how I got into plant virology, almost by accident.
Yeah, that's an interesting story.
Yeah.
So after you graduate from PhD, why you go directly to academia instead of other position?
I was about a year from graduating, and my research was going very well, had been very successful, and I had 2 children. I was married at the time, and I had 2 children, so I really was desperate to have a job that paid something. We were tired of living like in graduate student housing forever for 2 degrees. And I went to the library at Cornell one day, and I was sitting in the library looking at journal articles and in Science magazine, and in the back, I turned the page, and there was a half a page ad, a huge ad from the University of California, Berkeley, and the title was Plant Virus Vector Research. And they were looking for an assistant professor position in plant virus research to study vector-specific transmission of plant viruses.
So it's a perfect position.
That was my Ph.D. title, my thesis title. So I read the description of the job a couple times, and I thought, there is no way I can't get this job. Yeah, I just felt like this job was for me. So I went back to Dr. Rocco and showed it to him and told him I wanted to apply. He was not happy because he wanted me to do a postdoc with him after I graduated. I was going to graduate about a year from then, and he wanted me to stay and work more with him. And then I also had an offer of a postdoc at Rutgers working on phytoplasma and leafhoppers. So I did have a postdoc offer, 2 postdoc offers. But this job looked so good, and I went out to California and applied for it, and I was a perfect fit for the job, and they told me I had the job before I left campus. So, I just fell into a lucky situation.
Yeah. So what's the most excited part of your work?
That's a very broad question.
Yeah.
What is my work? It's mentally, the way I viewed my job was half research, half education, because I have the attitude, I'm a little bit old-fashioned, that I think universities are first and foremost and primarily educational institutions. So I always felt that teaching was a very important part of my job. So I've taught quite a few courses in my career, different types. I enjoy teaching very much, and I always felt that about 50% of my responsibility was for teaching, and about half my effort went into research, on the average, more or less, in terms of mental capacity and time that you think about what you're doing and planning. Research was very important, obviously, and I really enjoyed research. I viewed research as my hobby because I enjoyed doing it on my own. Teaching I felt as my vocation. That was why I was here, to teach. Yeah. So I find that the teaching probably was very exciting. At the same time, the research I did was extremely enjoyable, and that was also exciting.
Yeah. So what is your best story in your career you can share with everyone?
Wow, that's a difficult question. I think how I fell into plant virology is probably a very interesting story. I think I was very lucky in my career. This isn't exactly a specific story, but I was very fortunate in having 2 excellent faculty advisors. For my master's degree, I had a person named John Mitchell at Ohio University, and he was from Edinburgh. He was an organic chemist who began working with plant biochemistry, and he took me on as his student, and I was for most of the time I was his only student. So he really focused on my career development, and we developed some rather unique tissue culturing systems to study the histology and the development of roots from callus cultures with various hormone controls. So I thought that was pretty exciting. As a high school student, I was always interested in biology, and I always had science projects for high school science fairs and so on. So I always had it in my mind I wanted to be a scientist, and he made that possible.
Wow.
And he was an excellent scientist. So from the very beginning, I had a really good mentor. And then when I went to Cornell and worked for W.F. Rocco, he was equally good. Dr. Rocco was a excellent scientist who really understood how to design experiments. He was a world-famous scientist in his area, so that helped me in the future because he had a reputation and I became sort of his protege, and he gave me a lot of support and allowed me freedom. Once he understood that I knew what I was doing and he trusted me, then he gave me quite a bit of freedom and support to do what I wanted to do research-wise. So I developed more quickly than other students might have. That's why I was able to kind of skip a postdoc and go straight to a faculty position, because I already knew how I was going to do my research. And I'm one of the unusual people in that I finished my career doing the same kind of research I started my career with. So for over 40 years, I was working on plant virus systems with insect vectors. And so you can really, when you just work with one system, you can really get very deep into that system and understand a lot.
So can you give some advices for the budding scientists now, based on your experience?
Oh, I'm very good at giving advice, maybe too much.
You can give some suggestions now.
Be yourself. Be honest with yourself. You have to love your work. Do the research. Try to design your career and your job around something you enjoy doing. That's critical, especially for a faculty member. I think you just have to be scrupulously honest. To be a scientist, you have to expect failure. I always assume when I do an experiment, when I design I have a hypothesis, or I'm trying to solve a problem, a field problem in plant pathology. You have a hypothesis, you can design some experiments that you want to do. Okay, when you design the treatments to test your hypothesis, the first and the most important treatments are your controls. So always design good controls, negative controls and positive controls. Think of all the controls you possibly can have and decide which of those would be critical to and then also you have sufficient number of replicated treatments, so that when you finish the experiment, you design the experiment so whether it works in your view or not, you have an answer.
Yeah.
It's a yes or no. You don't want to have an experiment that just fails and you're not sure of what it means. You try to design it close enough and sharp enough and well-defined so that either it's going to prove one thing or it's going to prove something different. And make sure nobody can question that research, that result, based on your controls. Yeah. And I always try to think, okay, what questions can people ask if they don't believe it? You know, did you do this? Did you do that? And yes, you can say, yes, they tested this, we tested that. All the controls worked well, and therefore the results are true, whether you like them or not. The other piece of advice I would give, or kind of an insight into my thinking, is whenever I did an experiment, the first time, I expect it to fail. Because there are always unseen events and things you haven't thought of. So more often than not, or say frequently, an experiment will fail the first 1 or 2 times you try it, and you have to polish it and rework the treatments and do it over again. And don't be discouraged. My attitude was, if I go into it thinking it's going to fail, when it fails, I'm not depressed.
You know it will fail.
Yeah. No, if it doesn't fail, that's great. Yeah. But if it does, it's okay, because you kind of expected it to, and you can polish it and do it again. So I think a lot of students often become depressed or turned off to science because they want immediate results. And if things don't go well, they become depressed about it, because you put a lot of time and effort into something, and then you're depressed when it doesn't work. You feel like you've wasted your time, but you really haven't wasted your time. You've learned something just by doing it. You learned a lot, and then when you do it again, you'll do it better. So I never view that as a waste. You have to have the long view. Be looking at the long-term view. Another piece of advice I would give is when you're doing research, as a graduate student especially, but even as a professor I did the same, especially when you're younger, when you're walking around, when you have a problem, a hypothesis, you have a problem you're trying to solve, or you have data and you don't know what they mean, you need to be thinking about it a lot. When you're walking into work in the morning, maybe you should be thinking about the problem. And when you're walking home, you think about, what did I do all day? What does it mean? How do I interpret these results? What are the possibilities? And you keep thinking about it, and eventually over time you'll have an insight. You'll have an aha moment. You go like, oh my, I think this means this. And then you can test that. And I've had a few times when that happened to me, asking for stories that are interesting. When I was a grad student, I was doing electron microscopy on aphid salivary glands to look at viruses to find out how they were moving through the aphids. And I learned that the aphids come as, they were all females, and they either may have wings or not have wings. And I discovered that the aphids without wings, the Apterus aphids, had larger salivary glands. They were just bigger. So they were easier to work with and look at and dissect and so on. And so I wanted to rear a bunch of aphids to look at on the EM, and I only wanted to look at the non-winged ones. So I had an infected plant and a healthy plant. And it was infected with Barley-Yellow virus and healthy, and I put 3 or 5 aphids on each one, allowed them to make babies overnight. Then I removed the adults, caged it, and let the babies, maybe 30, 40, 50 aphids on each plant, go for 2 weeks until they matured into adults. Then I was going to harvest the aphids off of both plants and look at the salivary glands. I had the healthy aphids with no virus, and I had the aphids with virus, and see where the virus is. But I only wanted non-winged aphids, so I collected all the aphids off the healthy plant, and they were mostly non-winged because they were young seedlings and very healthy, and the aphids did well, and they were all non-winged aphids. I went to the other treatment, it was infected plant. They were mostly wings. They all had wings, except for a couple. And I thought, this is weird. Bad luck. So I did it again. And 2 or 3 weeks later, same result. And aha, whoa, this is not an accident. So then I set up a whole series of experiments with multiple treatments and replicates. And what we found was that aphids that were reared on infected plants developed wings. So this aids dispersal and aphid spread. So that was an interesting thing to find out, that the plants, the infected plants, are physiologically different, and they induced aphids to develop wings. And then from that, we did a lot of research, and we got 2 or 3 publications, and it also had an impact on aphid epidemiology. And so that was sort of an accidental discovery that just, you had to have your mind open and be looking for things, and I fell into that. So that taught me a lesson. Thereafter, you were always looking for these kind of meaningful results, even if it didn't work. See, essentially, it didn't. Initially, my initial experiment didn't work.
So the last question, if you didn't teach plant pathology and didn't work with plant viruses, what would you do as your career?
Do you mean within the university framework as a professor, or?
No, it's just in general.
Quite frankly, I've never thought about much else. I've always wanted to be an academic professor since I was in college. That was always my goal. And I think since high school, my goal was to be a teacher. One thing I might do more of was teach more. I might want to be a teacher because I like teaching young people and sharing what I know about biology with young people and getting them interested in science. So I really like that. Other than that, I might want to be a professional fly fisherman or a canoeing guide. I like the outdoors, and I think actually if I could have had maybe another area I might have gone into is ecology. I might like to have done more ecology and more outdoor type research or work of some type in the outdoors.
Wow, that's a different one.
Yeah.
Thank you, Dr. Gildow, for sharing all your experience with us.
Well, you're very welcome.